The overall goal of this procedure is to produce a 3D tissue-engineered model of the human esophageal epithelium for use as an experimental platform. This is accomplished by first isolating normal human esophageal fibroblasts and epithelial cells from human esophageal tissue samples. In the second step, the fibroblasts are seeded onto a de-epithelialized acellular porcine esophageal scaffold and cultured for 7 days. Next, the scaffold is inverted, and the isolated human esophageal epithelial cells are added to the culture for 4 days. In the final step, the model is moved to the air-liquid interface for a further 5 days of culture, allowing a mature stratified epithelium to develop. Ultimately, immunohistochemistry is used to characterize key markers of proliferation and differentiation expressed by the model epithelial cells.
The main advantages of this technique over conventional 2D cell culture is that it allows cells to have the cell-cell and cell matrix interactions that they would have in vivo. And using this technique, we can produce a mature stratified squamous epithelium similar to the normal esophagus.
Generally, individuals new to this method will struggle for 3 reasons. 1, it can be a challenge to ensure that the majority of epithelial cells do not differentiate before being included in the scaffold. 2, care must be taken to ensure that the correct air-liquid interface is provided during the period when the epithelium is maturing. And 3, it can be a challenge to maintain sterility throughout the culture period.